Recording begins. This is October 20th, 2025, Biology 105. Okay, as you all know, Canvas is not working. When's it going to start working again? We're not sure. This is great, all right? So we're still going to continue with lecture material as usual. We're still going to plan on continuing with lab material as usual. If the Canvas shutdown prevents access to an assignment that you needed to complete, then hopefully this will just be up and running again by tomorrow. But you reach out to me, or if it affects everybody, I will catch it and make a big announcement and adjust due dates, okay? But everything was due yesterday at 11:59 p.m., and my understanding is that everything worked until sometime after midnight. So I'm very grateful for that, at least. I'm sorry for the serious disruption caused by this, and though I have no power over it, I still feel for you. Let's go ahead and get back into our PowerPoint discussion. Unless there's any last-second questions before we start? I'd love to offer to say, hey, come over to my office hours today after lecture and see how you did on the lab quiz. We can discuss answers and things like that, but I can't look at your lab quizzes because they're all on Canvas. So maybe Tuesday, Thursday office hours, you come by, talk to them, I'll help you out. Either way, let's go ahead and get back into photosynthesis, and I want to wrap this and get into our week 9 material, which is cell communication. Okay, we have been talking about photosynthesis. Before that, we spoke about cellular respiration, right? You've seen now the overlap between the two in that both of them rely on a membrane's impermeability to protons in order to use high-energy electrons to pump protons to generate a proton motive force, which flows through ATP synthase to generate ATP. The fate of that energy differs between cellular respiration and photosynthesis. Remember that in photosynthesis, you have two sets of reactions. You have the light reactions, where light energy is harvested and converted into chemical energy in the form of ATP, and then reducing power in the form of NADPH. These outputs of the light reaction, light reactions, are then going to go into the Calvin cycle, where that energy and reducing power is going to be utilized to build glucose. I've already emphasized to you that there's actually this special molecule called G3P that gets spit out of the Calvin cycle, and it's the real output of photosynthesis. Then it goes through some other pathways to become glucose and other things. But for the purposes of any Bio 105 class that you take, this process makes glucose. All right? Cool. So major inputs and outputs here. Water is consumed by the light reactions. Water is oxidized, and that produces oxygen. You pull the electrons off of it, you get oxygen, okay? In the Calvin cycle, carbon dioxide is consumed, and it is reduced, and it is built up into these larger organic molecules. Questions about that stuff? Once again, these concepts are difficult, so you're gonna want to review these things. But hey, since I'm posting lecture recordings, I feel like it kind of frees me up to say, we covered it. You know? You can watch the video if you want to refresh it again. Pointing this out one last time. This is the big figure, the big money figure from photosynthesis, where this is what I like to really test on is the light reactions. I like to test on proton motive force, ATP synthase, the electron transport chain, because that process is similar between respiration and photosynthesis, and therefore I think it warrants, repeating, since biology thought it was important enough to repeat. Very fun. Cool. This is the other big money figure right here. So these two are the big money figures, where if you understand these, you understand photosynthesis to a decent degree. Cool. Linear electron transport chain, cyclic electron flow, so nonlinear electron flow, linear electron flow, nonlinear electron flow, Z scheme, electron goes up in energy levels. Questions? Okay, sick. Watch the videos, check it out. I ended with a big statement about Rubisco. This has resulted in so many deaths. I tried to get you all very excited about Rubisco, and I'm sure that it's all you thought about all weekend long. So now let's resolve what I'm actually talking about here. Okay, what is Rubisco? Rubisco is a very important enzyme. It's probably the most prevalent enzyme on the planet, most abundant enzyme on the planet. 50% of the protein in a leaf is Rubisco. That's going to determine nitrogen requirements of plants. And as you might know, nitrogen deficiencies in soils, or nitrogen overabundance in soils, or nitrogen supply, nitrogen fertilization, huge issue in agriculture. All right? And if half of the leaf protein, which is where you need the nitrogen, all right, is made up of Rubisco, well, then Rubisco is one of the main reasons that we need all this nitrogen fertilizer being made. So many things cycle around Rubisco, which is a joke because it's in the Calvin cycle. We're having too much fun today. Here's the name of the enzyme, which you don't need to memorize, but which does explain its importance, okay? Which does clarify an important aspect of it. So, ribulose 1,5-bisphosphate carboxylase oxygenase. What's that mean? Well, remember, the enzymes, they always tell you what they do, or usually tell you what they do in their name. If we look at the Calvin cycle, there's this enzyme called ribulose bisphosphate. It's ribulose 1,5-bisphosphate, actually, okay? Which tells you that the 1,5-bisphosphate just means that at carbon 1 and at carbon 5, there's a phosphate, and there are 2 phosphates total. Ribulose, a sugar, so it's ribulose 1,5, carbon 1, carbon 5, bisphosphate, 2 phosphates, phosphate 1, phosphate 2. That's the molecule, okay? That carbon is going to get fixed onto and drive this whole cycle, okay? So ribulose 1,5-bisphosphate carboxylase oxygenase means that this enzyme substrate is ribulose 1,5-bisphosphate, and it can carboxylate it or it can oxygenate it. So it can either attach carbon or attach oxygen. And that's Rubisco, all right? It starts the Calvin cycle. It combines ribulose bisphosphate with CO2 when it's functioning correctly, okay? But it can also, so this would be carboxylating ribulose bisphosphate, adding carbon to it. Question?
So the actual enzyme's use is just to be able to restart that cycle over by like adding carbon or adding oxygen?
Yeah. Well, its function is to attach carbon, but unfortunately, it can also oxygenate. And if that happens, then you ruin this ribulose bisphosphate. It's kind of like fermentation, where you take it and you make it into a useless byproduct.
Is that photorespiration?
It is photorespiration, yes, exactly. So normally, when the system's working correctly, this 5-carbon molecule gets an additional carbon attached to it from CO2, and then it immediately splits into 2 3-carbon molecules, and that sort of drives the whole cycle. That's the starting fuel for the cycle, and then the cycle runs and regenerates this ribulose bisphosphate. It's a cyclical process. They're annoying. There's too many steps, you know. But Rubisco is important because it's a very imperfect enzyme. While it should be attaching carbon dioxide to ribulose 1,5-bisphosphate, sometimes it will actually oxygenate, okay? It'll actually oxygenate the ribulose bisphosphate, all right? So this is the normal process here. The active site also has an affinity for oxygen, okay? That's because when Rubisco evolved, the atmosphere had hardly any oxygen in it. It was a world before photosynthesis, or at least photosynthesis as we know it now. So oxygen levels were really low. So the Rubisco enzyme evolved without needing to worry about oxygen being present. But then when it became a prevalent and abundant and functional, successful enzyme, it kind of screwed itself over because it made all this oxygen. But oxygen can also fit into the active site. And if oxygen gets jammed in there, then you get this process called photorespiration, okay? This causes CO2 to be released actually, and you lose fixed carbon, and you lose the ribulose bisphosphate, which took a lot of energy to build up and regenerate. So it's just a net loss for the plant. It's a net loss for carbon fixation. It's just a bad situation for everything involved. And there's no fun alcohol product generated by photorespiration that we harvest and use for other things. So it's all bad, photorespiration. It happened because Rubisco evolved in an atmosphere without oxygen. All right? And then Rubisco led to a mass extinction because it killed all these organisms that weren't used to high oxygen concentrations in the atmosphere. So Rubisco's been a really important protein for a long time. All right, it may be an evolutionary relic. It's an evolutionary relic. All right, when we talk about ancient things like this, we have to say maybe, but I'm gonna go out on a limb on recording and be like, this is an evolutionary relic. All right, photorespiration. It's especially a problem in hot, dry climates, okay? So remember we talked about stomata and how stomata open and how stomata close and how stomata are little pores in the plant that let CO2 come in and oxygen leave. Well, when plants are, you also lose a lot of water through these stomata. So when plants are really hot, they close their stomata, or when they're really dry, they close their stomata to slow rates of water loss. But here's the problem. Now you're still carrying out photosynthesis, so you're reducing CO2 levels inside the leaf, and you're driving up oxygen levels inside the leaf. And when is it the hottest and the driest? During the day. So that's when you can actually drive the light reactions, and that's when you can actually drive the Calvin cycle. So photorespiration becomes a serious issue. All right? But plants, not content to rest upon their laurels, have developed modifications to photosynthesis that are attempts to reduce rates of photorespiration. So photorespiration can reduce crop productivity by crazy amounts. Globally, sometimes those estimates go up to as high as like 70% of potential yields being lost because photorespiration is such an issue. Some plants have modifications to their photosynthesis, and I'm not going to really be testing on this because you should learn about this in your Botany 105 course or in other courses that, will go into more detail, but I really don't think it's fair to talk about photosynthesis and not talk about these concepts at all. A question, please. No problem. So C3 and C4 plants. C3 plants are the standard issue plants, standard issue photosynthesis that we've been talking about. Let's dial it back to right here. Okay, cool. Remember how Rubisco attaches CO2 to ribulose bisphosphate, and then you immediately get these 3 carbon molecules? It sort of takes a 5 carbon molecule, slaps another carbon, makes a 6 carbon molecule, then that thing splits into 2 3 carbon molecules. C3 plants, that's what they do. You get this 3 carbon molecule that is the first product of carbon fixation. In a C4 plant, there's a 4 carbon molecule that you make before you do this. So CO2 gets initially fixed onto a 4 carbon molecule, and so we call them C4 plants. All right? There are also CAM plants, and CAM plants also use that 4 carbon compound, but they just work slightly differently. All right? So you got the regular old C3 plants. When the CO2 levels get low and the O2 levels get high, they carry out a lot of photorespiration. In the C4 plants, they have these special cells where the Calvin cycle doesn't happen and there's no Rubisco. And in these, it's more accurate to say that they have sequestered Rubisco and the Calvin cycle into special cells. That's more accurate. So most of the cells can be in the leaf can take carbon dioxide and make this 4-carbon compound, which they then ship to another cell here, okay, where all the rubisco is. And then you just dump CO2 into that special cell, the bundle sheath cell. You just keep dumping CO2 there to increase concentrations of carbon dioxide surrounding rubisco. In CAM plants, you do the same thing. Well, you do a similar thing. You open the stomata up at night when the transpiration rates are lowest, and you temporarily store CO2 into this 4-carbon compound. Then when it's daytime and you've got all that ATP and NADPH to drive the Calvin cycle, you release CO2 without having to open the stomata. So both of these are designed to reduce photorespiration rates by increasing CO2 concentrations around rubisco when it's got the energy to drive the Calvin cycle. Question?
So, let me see if I'm understanding this right. They're essentially stockpiling for CO2 and it's like easier to get so they can use it for the Calvin cycle in the day.
Yeah, exactly. Exactly. You open your stomata at night, you're going to lose less water. So you open the stomata at night and then just stockpile the CO2 in these 4-carbon compounds. Now, if you think about our discussion of energetics, you immediately could be clicking here like, wait a second, I'm building a 4-carbon compound? That's an endergonic reaction. So energetically, this adds an additional step to the pathway. And therefore, if photorespiration is not a problem, C3 plants win. They're more efficient. But if photorespiration is enough of a pressure, then C4 plants and CAM plants can win. And let's take it one step further. One of the major issues that we talk about when we discuss climate change is carbon dioxide concentrations in the atmosphere. They're going up, all right? Due to increased decomposition rates, deforestation, fire, whichever thing you want to point to. There's a lot of reasons, fossil fuel burning, there's a lot of reasons that carbon dioxide concentrations are increasing in the atmosphere. But think about this, right? If CO2 is super abundant in the atmosphere, photorespiration is not going to be a problem. So plants that carry out C3 photosynthesis are going to have an advantage, and the advantage that the C4 and the CAM plants had in ecosystems goes away, because you don't have to have your stomata open as long to get the CO2 that you need during the day. And there are trackable, observable shifts in plant communities where C4 and CAM plants are becoming endangered in habitats where they used to be dominant. Fascinating stuff. A question at the back, and then I'll bounce down to you here. Yeah. Oh, really? Oh, how'd I do? Awesome, thanks. Yes?
I was supposed to just ask if that was like an observed thing, so
I got it? Yeah, it's observed. Yeah, C4 plants especially. There's a lot of these grasslands that are dominated by C4 species, and now with rising carbon dioxide levels, some communities are shifting more towards C3 species. Which, is it bad? Is it good? Is it case by case? That's a question for other people. We'll leave that for the ecologists. We'll leave that for the class that you take with me of plant ecology, maybe. I don't know, or we'll talk about it there. It's a fun time. Or don't. Whatever. It's all good. I hope you just all have a wonderful degree experience. What are we talking about? We're talking about different types of photosynthesis. Like I mentioned, this is not a huge focus, but it's interesting you gotta admit, it's a little interesting. And it really ties together all the photosynthesis talk, and you really shouldn't leave BIOL 105 thinking that there's only one photosynthetic strategy, because this rubisco problem, this photorespiration problem, has been around for a very long time, so it makes sense that plants would begin to evolve ways to deal with it. Anyways, oh yeah, CAM is Crassulacean acid metabolism, because the family is Crassulaceae, where it was initially discovered. They tend to be very succulent plants because they need a lot of central vacuole space to store these 4-carbon compounds that they generate at night. That's the last thing. That's it. Cool. You want to do one of these? We all love the flow charts. Let's do a flow chart. Okay. Photosynthesis converts what into chemical energy? Light. Light energy, radiation, light, all good answers. Photosynthesis includes two sets of reactions. Look, there's a membrane here to hint the one that is B. It includes what? Includes Calvin cycle here, light reactions here. Because remember, the light reactions require that thylakoid membrane. Sick, solid, awesome. The green pancakes. All right, let's focus now on the flow from light reactions. In the light reactions, water is split and what happens? That's what I'm looking for, yeah. Oxygen gets released. Exactly, okay? Water is split and oxygen is released. You could also say like, water is split and electrons are then elevated to a high energy state or something like that, okay? But I want oxygen is released. Okay, the light reactions in which light excited electrons of chlorophyll, so that's from that reaction center complex, right, where it gets kicked up to a high level and goes doo-doo-doo-doo-doo. The light excited reactions of chlorophyll are passed down, what do we call this? An electron transport chain, that is correct. Reduce, also, at the end of this, you use some of those high energy electrons to reduce NADP plus to what form? NADPH. Remember, P for photosynthesis, P for plant. It's NADH in respiration. Sick. The electron transport chain produces what by chemiosmosis? ATP. Let's zoom over to the Calvin cycle. Have I told you that the person who's credited with figuring out the Calvin cycle's name is Melvin Calvin? I've mentioned that. It's a great name, just a great name. Photosynthesis includes the Calvin cycle in which carbon dioxide is fixed to ribulose bisphosphate and then a 3-carbon molecule is reduced. You remember that step, right? The 3-carbon molecule is reduced using NADPH, exactly, and you need some energy from solid. What do you get from reducing that 3-carbon molecule? Yeah, G3P or glucose, you know, or glucose and other high energy organic molecules. Yeah, everybody feel okay about that? Awesome. I love the respiration and photosynthesis, like 1-2 punch, super fun, super solid sequence of things which everybody should learn from Bio 105. I hope that you found it informative. Let's move on to week 9 material. This is unheard of, people. We are in week 9 of Bio 105, and we are looking at the week 9 PowerPoint. I don't know if you realize what a big deal this is. On the Monday. It's nuts. Yeah, right? It's unheard of. And we still have like 22 minutes left in lecture. I know. We're going to make progress. There's only 51 slides here. Wow. I don't want to count my chickens. All right. So let's get into it. Cell communication, okay? Look, they have cell phones. It's like a joke. It's like a good joke, okay? So cell-to-cell communication, how do cells communicate? It's tricky because if you just relied on diffusion of signals from here to 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 here, it'd take forever. So we have other pathways where you can get a communication from, oh, I stubbed my toe, to me, you know, attacking this wall and tearing this whole room apart with my sheer strength, okay? You have to have a way to pass signals around quickly. Cell communication, all right? Not all of our discussion is going to be based on quick signaling, though. That's just what that little comic was meant to talk about. Not all of our discussion is about fast, rapid signaling from disparate or distal regions of your body, but also, how does a single cell detect a signal from outside, all right, outside of it, and then convert that signal into a cellular response? Cool? So one of our big things is going to be like, you've got signaling molecules going through your body, right? You've got a lymphatic system where you have all these hormones that are being pumped out of different glands and they move through your body and tell a bunch of cells to do the same thing at once. But how do the cells detect that signal, okay? So a lot of our cell communication discussion is going to be on proteins, and it's going to be on receptors and signal reception and transduction pathways. Okay. We'll talk a little bit about the different types of signaling. All right, we won't get into too much detail there because we're going to work a lot on talking about receptor ligand complexes and how signals can be amplified and how we actually elicit a response from within the cell. Also, we got to learn the three main types of membrane receptors. If I'm being honest, this is probably the driest part of the entire BIOL 105 lecture from my end. It's just like you have to memorize three things, three types of receptors without there being a lot of, there's just not a lot of justification for, why would I memorize the G protein coupled receptors? And it's like, well, someday you'll learn more about them. It'll be good if you know how they function. Anyways, phosphorylation cascades, it's going to be great stuff. It's going to be great stuff. Cellular messaging, let's do it. Let's get going. Here we go. Cells communicate. How do they do it? What processes are essential for cell communication? What processes rely on cell communication? Okay? So, we've got recognition, development, mating, responses to the environment. What does any of this mean? A cell in your body needs to be able to identify and recognize if another cell is part of you. Makes sense, right? You've got your immune system and all of this as well on top of that, and you need your immune system, to be identifying that your cells are not to be targeted for destruction. So cell-to-cell recognition, super important. Development. You started as a single cell, and then that single cell divided a bunch, and it became this big, complicated, multicellular organism, all right? During development, how does any of that happen? Through cell-to-cell communication. Basically, the thing gets bigger, and then it kind of competes, the cells compete with expressing different signaling molecules, and you get this weird banding pattern that gets set up in the developing embryo, and then that leads to different development in each band, which then releases different signaling molecules. You'll talk about it in developmental anatomy if you go that route. It's super interesting stuff, but I just want you to recognize that, yeah, during development from a single cell to a multi-celled organism, there needs to be some sort of communication between cells. I'm going to be a hand. The rest of you, don't be a hand. I'm the hand, all right? That sort of thing. Solid. Mating. Gametes need to be able to recognize different gametes. Cool? Responses to the environment. Single-celled organisms need to respond to the environment. The multi-celled organism needs to respond to its environment. And that response is mediated by by changes in activity of cells. Fair enough? I love it. So why does this, I breeze through this a little bit aggressively because it's one of those things where you're like, you probably understand that cells you need to be able to regulate what's going on inside a cell. Okay, here we go. Some different categories of signaling. There's gap junction signaling. Gap junctions are like plasmodesmata, but for animal cells. They're little protein complexes where it's a channel that joins the cytoplasm of two adjacent cells. So you've got a little channel there that's going to make it's going to make it very easy for this signaling molecule to diffuse into the target cell. But it only works with adjacent cells or a bunch of cells in a long chain or a sheet of cells where you're relying on the molecule diffusing. It's still going to be diffusing, right, this signaling molecule. Gap junctions, okay? Let's continue moving forward. Your autocrine system versus the paracrine system. In the autocrine system, a cell makes a signaling molecule, it releases that signaling molecule, and it detects that signaling molecule. All right? So it's kind of signaling to itself. This can be modified to have a bunch of individual cells all releasing the same signaling molecule. So you're receiving your own signaling molecules as well as adjacent cells' signaling molecules, which sort of straddles the line between the autocrine and the paracrine system. in the paracrine system, a signaling cell releases a signaling molecule, which then diffuses and reaches target cells. Cool? Now, in the endocrine system, okay, which is, I said lymphatic system earlier, and I really meant to say endocrine system, but I said lymphatic, right? Inaccurate. Mistake. It's on recording. I misspoke. Yeah. The whole class ends. You all get whatever grade you have right now, and we're just leaving. Endocrine system. All right, sometimes a signaling molecule gets jammed into the bloodstream, and then it moves throughout the body. This is faster, right, than relying on diffusion through the extracellular matrix and just diffusion throughout sheets of tissue, or diffusing from here down to your legs. Put it into the bloodstream, then you can diffuse it throughout the body much more rapidly, okay? So that's the endocrine system right there. Solid? Question, please. Sorry, littler? What does a signal... Totally. It can do any number of things. We will talk, that's going to be the really frustrating part of this cell signaling discussion. Usually, here's how I teach it. The signaling molecule the signal is received by a cell, and that causes the cell to change gene expression in some way. The nature of the signaling molecule, it can be different things. Sometimes it's a lipid, sometimes it's an ion, all right? It all depends on what receptors are there to receive the signal. So the general term of signaling molecule just covers anything that signals other cells to do something. And the something that they usually do is change patterns of gene expression or regulate metabolic pathways. Solid? Sick. Okay, I love it. Yes?
Really quick, can you explain what gap junction is again? I kind of missed it.
No problem. Let me draw a picture. Yeah. Yeah, totally. Oh yeah, it's just in the name. Yeah, there's two cells. They're pushed up against each other, and there's this little protein tube that joins the two adjacent cells. Huh? Yeah, basically. Yeah, it's like a channel protein. Yeah. And then that gap junction just allows things to diffuse. Now, the gap junctions are also tightly regulated, just like the plasmodesmata, but that's something for like a cell bio class to get into. For us, we're just going to consider it as like a little tube. Of course, solid. Okay, moving on. So you've got local signaling, you've got long distance signaling, and then you've also got synaptic signaling. So the local signaling, this is all of the autocrine, paracrine, gap junction. It only works with cells that are nearby, or it takes a long time to get that signal transmitted. The long distance signaling, okay, works for things that are more distant, okay, that does make sense. You pump the signaling molecule into the bloodstream and you can distribute it throughout the organism, all right? And then when you need a really rapid signal to be delivered, you use synaptic signaling, okay? And synaptic signaling is when you're using neurotransmitters, it's when you're using your nervous system. So I'm triggering all of these rapid little movements of muscles. I can receive sometimes a signal through my eyes that makes me flinch or go back, knee-jerk responses, right? Reflexes, things like this. So when you need a signal to be transmitted very rapidly, diffusing the signaling molecule through your bloodstream isn't fast enough, all right? If I have to dodge because somebody threw a shoe, I need to do that a lot faster than if I was waiting for the blood to circulate to all of my arms and legs, and then it would make me move like this. It's no good. I'm getting beamed with the shoe. It's no good. It's no good for me. Okay, so synaptic signaling requires these special nerve cells that essentially transmit electrical signals. It's all about proton gradients, just like everything, okay? It involves sodium, potassium ratios. It's something to be covered in a course that can really delve into it. But for our purposes, synaptic signaling, you've got these long nerve cells that can transmit an electrical signal and just be like ding, and it's like zip it right there from the start to the bottom. Then what happens is at a synapse, which is the point where two cells, it's the space between two cells, all right? At a synapse, the nerve cell just kind of dumps out neurotransmitters, all right? And neurotransmitters, once they leave the synapse, they're just hormones, all right? Neurotransmitters and hormones are basically the same thing as signaling molecules. It's just that you can more rapidly trigger their release near their target cell using synaptic signaling than using the endocrine or hormonal signaling pathway here, where you rely on movement through the bloodstream. So clearly these are going to be used in different situations. Question? No? Okay, cool. So yeah, there's so much to talk about with this stuff. If you ever really want to learn if you like the practical, how does this affect me sort of biology, I cannot recommend enough. The lecture's free now. You can find Robert Sapolsky's lecture on the biology of human behavior, all right? It goes to like a 300 level biology understanding and ties everything to human behavior, all right? So it's great. You learn about neurotransmitters and hormones and these different types of signaling beyond the level that we do here. He wrote Why Zebras Don't Get Ulcers. It's a lecture recorded at Princeton. It's like a cool, fancy thing. Stanford, maybe? One of those. One of the fancy spots. Anyways, I recommend it if you want. If that was like, oh, I want to talk about that all lecture, check out that lecture series. It's good. Okay. Here's sort of a fun example of cell signaling. This is autocrine/paracrine signaling. All right? Check this out. This is quorum sensing in bacteria. So you've got all these bacteria living in your body that aren't trying to kill you yet, because they know that if they tried, they would lose, okay? But if there were enough of them, they might be able to get you. So there's this thing called quorum sensing, and a ton of bacteria do it, where they're just exuding these signaling molecules, and they're all doing it. And if the concentration of the signaling molecule gets high enough, then they switch on these genes that make them pathogenic. And then they're going to try to get you, all right? So quorum sensing, what does it really do? It lets you know when there's a lot of other individuals around you. A quorum is a term that you might have heard when you're voting for something. If you have a majority enough to secure a vote, you say you have a quorum, okay? So this is the same terminology, but this is the bacteria saying our population is high enough that we need to change our gene expression to either try to kill the thing we're living in, or maybe to start to form like a biofilm or specialize in our functions a little bit. It's quorum sensing. It's cool, but it's an example of autocrine and paracrine signaling. Awesome. Synaptic signaling, yeah, you get the electrical signal, zap, you release the neurotransmitter, rapid diffusion to the target cell, boom, response. Cool. Now, here's the similar pathway for all of these different types of signaling, okay? No matter what, eventually a signaling molecule reaches a receptor on a cell, whether that signaling molecule originated from within the same cell or from an adjacent cell, or it got dumped out of a nerve cell, okay? No matter what, or it got dumped out of the bloodstream and surrounded the cell. Eventually there is a signaling molecule which hits a receptor. The receptor, all right, transmits the signal, and that signal changes forms. The signal is transduced, okay? It goes down what we call the signal transduction pathway. So the signal changes form. What do I mean? Look at this. The signaling molecule never crosses the phospholipid bilayer. That means that this signaling molecule Well, it must be hydrophilic, large, charged, something like that. If it was really small or hydrophobic, it might be able to bypass this pathway and move into the cell. Remember steroid hormones and how they are all actually lipids? They're like cholesterol molecules. Steroid hormones, they diffuse across the phospholipid bilayer because they're lipids, all right, and can elicit a response inside the cell themselves. But in this pathway here, check it out. The signaling molecule never enters the cell. So here's the signaling molecule. That is the first form of the signal. A receptor molecule receives that form of the signal and transduces it into another form of the signal. Maybe by phosphorylating a molecule and making it active, all right? There's all sorts, usually that's the pathway that we'll be talking about. But it's going to, maybe it produces a bunch of, it breaks something down, like it breaks down a lipid, a phospholipid, and releases a piece of it, and that's a signaling molecule. But now it's inside, and the signal has changed forms. We'll talk about some examples. But the key thing here is that if the signaling molecule doesn't enter the cell, then for the response, which originates inside of the cell, to occur, the signal needs to change forms and get passed into the cell. Does that make a little bit of sense? Okay, so this is the standard sort of signal transduction pathway with 3 stages. 1, signal reception. A receptor protein receives the signal. 2, signal transduction. That receptor protein transmits the signal in a few different forms, okay? And then at the end of that signal transduction pathway, some cellular response gets activated. Often it is through changing the expression of genes. So you get a new enzyme, or you produce something that inhibits an enzyme, all right? That's usually what's going on. Is all enzyme-based, you know. Not every time, but yeah, I don't have to worry about that. So check that out. We've got the 3-step pathway, the 3 stages of cell signaling. Only works when the signaling molecule does not enter the cell. So there can be cell surface receptors that work in the way that we just spoke about, or there can be intracellular receptors. And intracellular receptors, they don't need necessarily that whole signal transduction pathway because the signaling molecule can diffuse across the phospholipid bilayers of both the outer membrane and the nucleus, and then can elicit the cell response directly. It still interacts with some receptor, okay, so it's not directly eliciting it maybe, but the receptor binds to its ligand, and then that thing can pretty directly interact with the genetic material and affect gene expression, all right? So we're just looking right here. There are some receptors that are on the surface of a cell, and there are some receptors that are inside of a cell, and whether the signaling molecule interacts with a surface or an intracellular receptor all comes down to the solubility of the signaling molecule and whether or not it can cross phospholipid bilayers, which I've been talking about since the very beginning. And that's where we're going to end today's lecture. I know I've got a couple of questions from folks, but you know, we're wrapped, so come and talk to me right here at the front and I'll answer them for you. Thank you for watching this recording.